Hello everyone, this is Anna and we're going to be doing the very last of the video lectures. This is part 2 of the special senses where we're going to focus on some of the basics of ear physiology. Alright, as normal, you've got your note slides which you can find in your PowerPoints. And those are all listed in here, and just make sure you know everything that's in here, okay? Next should be some lovely pictures, which we're going to focus on. All right, so if you remember from gross anatomy, we talked about the outer ear, the middle ear, and then over here is the inner ear. Okay, you need to remember that there is no communication between the middle ear and the inner ear that is direct. No fluid, air, anything goes through it. Instead, vibrations are going to come here. They're going to bounce on the tympanic membrane. They're going to bounce through the bones being transmitted. They're going to go through a membrane and then come into this area and go through the cochlea for sound. So this is the cochlea, which is served by the cochlear nerve. The vestibular nerve goes to the vestibule and semicircular canals. This is not where you do sound. This is where you do balance or equilibrium. Also remember the eustachian tube right here, which goes down to the back of the nasal cavity. All right, let's focus in next on the cochlea. We're going to slice it like this to look at it, the inside of it. All right, if you look over here, you'll see the cochlea and you can see that they've sliced through it right here. And what you're going to see are these triple chambers right here in each tube, and these areas are called the organ of Corti, okay, specifically when you get into the middle one. So what you have is this region right here, and then you have the scala vestibuli, the scala tympani right here, and then the scala media here, and then right there is where you find your organ of Corti. We're going to take this organ of Corti and we're going to blow it up right here. And we're going to start looking at the structures in more detail. Actually, let's flip to another slide where I've got it bigger. Okay. So now that you're basically all together on what the organ of Corti is and where you find it, you know that you're in your scala media. So what you have is your basilar membrane. I have the endolymph right here. And let's see what's next. You got the tectorial membrane, which has these little hairs sticking out. These are stereocilia. Now, this is not actual cilia. This is a bad name, all right? Not cilia. These are actually microphyllae, but we call it stereocilia. But it's not stereocilia, it's microphyllae. So these are folds in the plasma membrane that attach to what we call hair cells, okay? Which is a stupid name, but that's what it's called. And this is the receptor for hearing. When sound travels through the liquid of the endolymph, it bends the stereocilia that causes a mechanical change in shape. And that is going to trigger depolarization. And that depolarization will be picked up by the cochlear nerve and then travel back in that direction, okay? And that's basically how the organ of Corti works. Let's look at a pretty picture next. And I throw this in because I like the way they do the coloration. So right here, you see the organ of Corti, and you can see how they've colored the different tissues so that you can see the pathway that the nerves are taken, and you can see the lining of the tube right here, and then this would be filled with endolymph in here, okay? And then they've done the same thing over here so that you can see the stereocilia, which remember are microphyllae, kind of pooking up, and then those are attached to the receptor cells, which are called hair cells, and so forth, okay? This is the next slide, and all this is doing is giving you the pathway that you're going to memorize. This is supposed to be an arrow, didn't import correctly, okay? So basically what you've got is you got the vibrations of bouncing on the tympanic membrane, goes through the auditory ossicles, through the oval membrane to the cochlear fluids, The fluids move, bending the hair cells, opens ion channels, starting a local or graded potential, and that can potentially become an actual potential that goes along the cochlear nerve to the brain. That's it. Just write it out a few times and you've got it. All right. Now, another thing that's interesting is how you hear high-pitched sounds versus low-pitched sounds. We're going to look at this in a picture in a second. But basically, things that are close to the window, when they trigger those hair cells, your brain interprets it as high-pitched. Low-pitched wavelengths travel further. And so when they finally get to a cell right up here and they bend those hairs, they are much further away from the oval window. Okay? Let's actually look at a picture of that next. All right, so here you are with sound coming along. It bounces along the tympanic membrane that vibrates through the auditory ossicles. It's going to go through the oval window, transmitted across this membrane into the liquid of all of the scalas, the whole organ of Corti mechanism. Let's not worry about that too much. And then it's going to travel here. And depending on how big the wavelength is, whether it's like this or like this, it's going to go either less far or further. So this one, the wavelength traveled to here. And that is where it triggered depolarization of the hair cell. Other ones might go further and go all the way around to here where it gets deadened at the round window. Okay, and that is what determines the pitch of the sound. Okay. Let's look at a different version of this. I like this picture better to kind of convey pitch differences. So here we've got the sound coming through here, and you can see this is a very high-pitched frequency, and it depolarizes there at the organ of Corti. This is a medium pitch frequency, and it goes further along, and it depolarizes there, whereas low pitch goes much further and depolarizes right there. And that's kind of cool when you think about it. Okay? All right, let's look at transduction. All right, here's my note slide for transduction of the sound. So sound transduction. So remember, stereocilia or microphyllae. And you're just going to go along this pathway, write it out once or twice, you'll have it, okay? So if the wavelength gets to a part where they can bend the stereocilia, that causes a mechanical change in shape, potassium, potentially calcium, comes in, opens mechanically gated ion channels. So these are not chemical gates, these are mechanical gates. If that happens, you start a graded potential and you have exocytosis of glutamate, okay? And then those neurotransmitters stimulate the sensory fibers of the cochlear nerve, okay? Basically how it works, it's the same way with light, it's the same way with other types of transduction. All right, let's talk about equilibrium, and I'm sorry, when this converted from PowerPoint to PDF, it did some funky stuff with arrows, and I don't know why. So what I want you to know is that we're dealing with the vestibule and the semicircular canals when we're looking at equilibrium, which you can also think of as balance. This is called the vestibular apparatus, okay? Now, you don't actually just use this for equilibrium. We have three parts. You've got the vestibular apparatus. You have visual info. And you also have in all of your joints proprioceptors. All of these things are what ultimately helps your brain decide whether you're balanced or not, what your equilibrium is, and what's your orientation to the planet. That's why when you're driving, sometimes people get car sick and you tell them to look out the window far away, and that'll help calm down their stomachs, okay? That is why even if you have your eyes closed, you can tell if you're upright or not upright, because you can use proprioceptors and this information to figure out where you are in space. So all of these things are used together. And if one part is messed up, it can make you feel sick to your stomach and feel dizzy and get vertigo and all of that kind of stuff. Now, what do I want you to memorize? I want you to memorize dynamic equals semicircular canals, and this is an angular rotary movement, like when you spin, okay? So just write that out Dynamic equals semicircle equals angular rotation, okay? Then I want you to write out static equals vestibule equals up, down, forward, backward, all right? Very simple. Write it out, memorize it. All right, I have another notes slide, which I'm going to quickly go over, and then we're going to look at some pictures. All right, so with the vestibule, I do want you to memorize the otolithic membrane and the otoliths, and make sure you understand how they work for triggering the little hairy thingies that are going to cause transduction, okay? Let's look at a picture now. All right, so here we're dealing with the vestibule. All right, so vestibule, hold on a second. What do I want to do? Okay, I know. Vestibule equals static equilibrium, which equals up, down, or forward, backward motion. Okay? So the vestibule is a little bit different in that it has what is called an otolithic membrane, which is made out of a gel. And embedded in that otolithic membrane are these little crystals called otoliths or ear rocks, okay? So they kind of sink down into that membrane just a little bit, and they anchor themselves in, okay? And when you tilt your head forward or backwards, these rock back and forth. When they do that, they move the stereocilia. When that happens, that causes mechanical gates to change shape, triggering a local potential or action potential, which if significant enough will transmit here and then potentially generate an action potential. All right, so that's the vestibule. Ear crystals are real. Let's look at semicircular canals. All right, semicircular canals, okay, so that's this part right here. They are filled with the fluidy perilymph stuff, the endolymph stuff. The perilymph is up around it. The endolymph is the actual inside fluid. Sorry. So it's a liquid just like in the cochlea. And basically when you rotate your head and you spin, you slosh the fluid like in a cup. And that causes these hairs to go back and forth, bending the stereocilia, triggering mechanical gate changes, opening them up to release neurotransmitters, to then potentially depolarize the vestibular nerve right here. Okay? So remember, semicircular canals, this is dynamic equilibrium, which is going to be rotation of the head. All right, let's look at what that means by rotation. All right, so right here we have a figure skater, and she is standing still, and you can see here are the little stereocilia. This is filled with the endolymph. She begins to spin, and that causes the fluid to slosh in one direction. That causes these to all go that way. That causes mechanical gate changes. It transduces, and then you get an action potential. And then she stops, and everything sloshes the other direction, and it goes back the other way. Okay? And that is basically how it works. Okay? All right, that is the end of my presentation on the Ear Physiology Basics, and it was super basic, but I think everybody's got enough to go on for this course, and we can call it done.